A warm good morning, dears. Our next video is on translocation of solutes in plants. As we all know, plants transport their food materials and water from different parts of the plant to other parts of the plant. Example, if food is synthesized in the leaf part using chlorophyll pigment through photosynthesis, that is sucrose, that should be transported to all parts of the plant like stem, root, flower, fruit, etc. Similarly, the water that we give in the soil that has to be transported to all other parts of the plant like stem, fruit, leaf, flower, etc. Along with all these things, it should have some nutrient content like elements, etc. So, how does these things happen in a plant? So usually these ionic exchanges, ions in the form of minerals in the form of ions, they are transported through, they are considered as food and they are transported through phloem. And the other thing, water, that is transported to xylem. So the movement of organic food materials or solutes from one place to another place is called translocation of organic solutes. So these are translocated, the food materials are translocated in the form of ions. So in lower green plants like Eulothrix, Chlamydomonas, etc., the food is synthesized in each and every cell and there is no need of translocation. But in higher plants, they are synthesized in green parts and they has to be translocated to the other parts of the plant. So usually the foods that are synthesized are stored in the form of insoluble forms. They get converted into the soluble forms and then they get transported to next parts of the plant. Translocation of food materials that can happen in a plant in three ways. That is through by the way of direction. In three ways, there may be downward translocation. Example, from leaf to root. Synthesized sucrose or the food materials in the leaf part through photosynthesis is translocated to the root part where it doesn't photosynthesis doesn't happen. The other way is upward translocation. Example, from the root to the other parts of the plant like leaf or the leaf to the fruit tip of the plant, etc. for the development of bud, meristems, like that. So those movements are from down to up, so it is upward translocation. And another translocation is lateral translocation. That is from the xylem and phloem to the nearby lateral cells and up to the bark area. So that is lateral translocation. So the path of translocation in higher plants is usually xylem and phloem, as we said earlier. Xylem are dead cells and hence ascent of sap happens there, and the cells involved are tracheids, vessels, fibers, parenchyma cells. And vascular system consists of other live cell path of translocation, that is through phloem, where food is translocated. The ionic exchanges happens like ions, elements, hormones, etc. moves through phloem. It consists of components such as seed tubes, companion cells, phloem fibers, phloem parenchyma cells. The downward translocation of food was proved through ringing experiment. In this experiment, a plant was considered where it was kept live by removing a phloem region, a small region of phloem in the stem, and after a few days it was noted that the area above the cut showed a bulged region. So this bulged region was rich with food materials, sucrose, hormones, etc. And after a few days, rooting was noted from that bulging region. This indicated that through xylem, though water transportation was translocation was happening, the plant was alive using the water, and the food translocation from leaf to other parts of the plant, that was to the root through downward movement, was blocked by the removal of the phloem region. Hence, it got accumulated, and there, due to the hormonal activity, the rooting was induced naturally and the roots started to sprout. So this indicated that the food materials are translocated through phloem in downward direction. Another experiment was done by Dixon to show the upward translocation of food. In his experiment, he considered three different woody stems where he removed the leaves and phloem in first case, and in second case leaves and xylem was removed, and in the third case leaves was removed where the xylem and phloem remained normal. And after a few days, these three plants were um, dried, cut out and dried, and then the dry weight was considered. Where it was noted that the leaves and phloem was removed, it showed the lowest dry weight. This indicated that the plant was not able to synthesize the food since no leaves was there, and since the phloem was removed, no translocation of food was also done. Considering the other two plants, since xylem was present and phloem was present, the presence of the phloem itself indicated it could translocate the food from root to other parts of the plant, and hence the plant had higher dry weight. But since the phloem was removed in the first case, it couldn't translocate the food material, and hence when the plant was taken for experiment, what was the dry weight for itself? That itself was its weight. But in other cases, it had translocation process through phloem, and that showed the higher dry weight. So from this experiment, it was clear that the upward translocation of food materials also happens through phloem. So these two experiments showed that plants can have a two-way translocation, upward to downward or downward to upward. Two directional, bidirectional food flow could happen in plants through phloem. Translocation. The sucrose that is produced in the leaf part of the plant should be translocated to the other parts. Similarly, in some dry condition or in some rough conditions where water availability is low or when the plant is stressed, the stored food materials will be utilized by the plants for its sustenance. So in such cases, it should be translocated from the stored region to the whole parts of the plant. So these translocation, we consider two regions where the plant produces or stores the food material, or the region from which the plant requires the food material, that is considered as the source, and the region which requires the food materials is considered as the sink. For example, if we consider leaf as a source, since the leaf produces sucrose or food materials through photosynthesis, the produced sucrose materials through photosynthesis has to be transported to all other parts, example, to the root, where it can be stored in the form of tuber like carrot or potato. In that condition, the leaf is considered as the source, and the region, that is root, the carrot root or tuber, where it has to be stored is considered as sink. So the region of production is considered as the source, and the region of utilization is considered as the sink. So in this condition, we say a unidirectional, bidirectional, or uh, upward and downward movements, etc. All these things happens in phloem. But in xylem, only one directional, unidirectional water movement happens, that is from root to all other parts of the plant. So these happens in the phloem consideration, like food material transportation. The sieve plate take part in this process. The sieve plate, as we all know, that is a long column with holes at their end walls, which the end walls it is considered as sieve plates, and the they are each hole is connected through cytoplasmic strand, and hence it flows from top to bottom, and the whole sink to source to sink concept is completely based on the pressure flow hypothesis or mass flow hypothesis. So let me see what is it. So the pressure flow or mass flow hypothesis was proposed by Munch in 1930, and it was later elaborated by Crafts in 1938. So as per the image nearby, these are sieve plates which consist of holes. The both ends consist of sieve plates, and in between these each holes, one hole to the other hole, top sieve plate and bottom sieve plate is connected through thin sieve cytoplasmic filaments through which all this flow happens. So this is the structure. So this process consists of 5 steps, that is loading, osmosis, bulk flow, unloading, and again osmosis. So let me see it in uh, elaborated form. The loading, initially the loading process, where the leaf, here an example is considered from leaf to root. So source is considered as the leaf and sink is considered as the root portion of the plant. So the leaf consists of sucrose, which is produced through photosynthesis. It will be transported to the nearby cells and it reaches the companion cell and then to the phloem. So these flow happens through active transportation by utilization of ATP molecules, and this increase in ionic concentration, sucrose concentration in the sieve cell, this in phloem cell, this increases the hypertonic condition in the phloem. So hence, to tally the hypertonic condition, the water from the nearby xylem cell moves to the phloem cell through osmosis. And since the water enters into the phloem cells, it increases the turgor pressure of the cell, and this turgor pressure pushes the water to the nearby cell. So along with this movement of water, all these sucrose elements will also be moving. Like if we pour a water in a tube, along whether we mix sugar or colored solution, whatever it is, the water alone will not be moving. It will be carrying all the sugar contents, all the colored materials, etc. along with it. Similarly, the water, when due to the turgor pressure, the pressure buildups, and that pressure will be pushing water to the next cell. So along with the water, this ions will also be moving. So this will be continuing, and this continuing to the region of nearby cells of zinc. So this flow, along with all the ions what is having in it, that flow is called as bulk flow. So after this bulk flow, the reverse happens, that is through unloading process happens, where the ionic concentrations will be moving towards the zinc, that is companion cell, and then to the nearby cells and then to the root. And this happens through active process similar to that of loading. The unloading also happens through the active transportation or active flow. Then after the movement of sugar or sucrose molecules to the companion cells and to the zinc region, these cells, the phloem cells, will be devoid of the ions. So hypertonic solutions will be formed. So instead of due to avoid that condition, to tally that, the higher amount of water molecules which is present in this, lower concentration of sugar will be there, higher concentration of water will be there. So in that condition, the water will be moving through the to the xylem, nearby cell of xylem, through osmosis. This flow continuously happens through, and this cycle continuously will be happening. Flow of xylem, water from xylem to the phloem, and then the bulk flow. Then as the sugar compounds will be moving out of the cell, the water will be increased in concentration will be increased, and hence the water will be moving back to the xylem. This continuously happens, and thus the flow of ions or sucrose or hormones, whatever it is, it will be moved from the source to its sink region. This can also be explained through a small experimental model, demonstration model, where as in the image, a beaker or a tub is considered which is having water in it, and a chamber A and chamber B is there, which is connected by a T tube, where chamber A is filled with higher concentration of sugar solution and chamber B is with uh, pure water or diluted sugar solution. In that condition, if we keep it in the water, what happens is this chamber A and B is made up of semi-permeable membrane, like the plant cell. So this semi-permeable membrane, the osmosis happens that because chamber A consists of higher concentration of sugar solution. To tally the amount of water, the water from the tub will enter into the chamber A to tally the sugar solution concentration. And since the water enters into chamber a turgor pressure will be developed, since it has a capacity. Above its capacity, when water enters, the water will be pushed to the nearby available space. So here the available space is the T tube, and this water will be moving towards the chamber B. and since here the sugar solution concentration is really low, so in that case the sugar solution will be moving towards chamber B, and here the concentration will be slightly rising. As the water get enter into chamber A, the sugar solution concentration will be rising in chamber B. This happens until the chamber A and chamber B becomes equilibrium, or the both solutions inside it becomes isotonic. When we add sugar solution, concentrated sugar solution to chamber A, this flow will be continuing. So this indicates, or this also demonstrates this is a demonstration model of mass flow hypothesis. This experiments or this proposal of mechanism consist of merits and some of the demerits or some of the objections we faced. Let me see what they are. The merits are like when a woody or herbaceous plant is griddled, the sap contain higher sugar that will be exudated from the cut ends, and positive concentration of gradient disappears when the plant is defoliated. These are the merits of the mechanism explained by Munch. And what are the objections or demerits he faced is that this hypothesis explains only unidirectional movement of solute, that is from source to sink. The backward movement is not explained. Then the osmotic pressure of mesophyll cells and that of the root hair does not confirm the requirements. Some other requirements are also there. How does it move correctly to the root region? The movement of the how does that pressure created towards the root or towards the source? Some of the osmotic pressure of mesophyll cells is completely not explained or included in this bulk flow hypothesis. And this theory gives a passive role of sieve tube of the protoplasm, but not an active part. So we say that without the using of ATP molecules, the bulk flow happens. But there is also evidences that bulk flow, the transportation can happen through active transportation also, translocation also. So that part was not explained in this mechanism. So by through this transportation of or translocation of solutes, the organs which are storing the food materials are like some of the examples, like leaves. In leaves, like onion, garlic, cabbage, the leaves contain the stored food material. Considering the roots, carrots, radish, turnip, potato, etc. Stem, it is turmeric, ginger, potato, etc. Then flower, it is cauliflower. Seeds like rice, wheat, corn, etc. And in forms, what are the forms of food that are stored is like they are formed in the form of stored in the form of starch, sugar, insulin, hemicellulose, oil and fat, and then protein in respective plants. So that's all. Thank you.